Today our competency number is 2.8, 2.9, 2.9. Platelets, structure and function. Then we will be discussing the hemostasis, the mechanism of hemostasis. It has three stages or three phases. In today's class, we will discuss stage 1 and stage 2. At the end of the stage 2, we will find how platelet plug is formed. Temporary hemostasis is made. Thereafter, this temporary hemostasis, temporary platelet plug is converted into clot. That is the stage 3. This stage 3 is done by two separate ways. There is intrinsic path, there is extrinsic path. By both the path, we get ultimately the final clot. After the formation of clot, within few hours, clot will be retracted, and then repair of the damaged endothelium will be starting. When it is repaired, then the process of hemostasis is completed. So, first phase, second phase, I will be discussing today. Your questions in different exams, it may be like that. Describe the process of temporary platelet plug formation. That is, you have to describe phase 1 and phase 2. So in the coming class of Saturday, in that class, I will discuss the third phase. That is how permanent clot is formed from temporary platelet plug. Is that clear to you? Okay. So let us start with platelets. Because these platelets are the key factor that control that is involved in hemostasis. So let us first study about the platelets. It has got another name. What is the other name? Thrombocytes. Good. It is one of the blood cells. There are three kinds of blood cells. Erythrocyte, leukocytes, and thrombocytes. Whose number is maximum? Who are abundant? Erythrocytes. They are in million. And platelets, they are in lakhs. What is the normal range? Right, 1.5 to 4. 4 lakhs per cubic millimeter of blood. When their count is less than 1.5 lakhs per cubic millimeter, then what is the condition called? Less than normal. Thrombocytopenia. So it is called thrombocytopenia. We will discuss or we will mention in what conditions. What are the conditions in which thrombocytopenia is seen? There can be thrombocytosis. What is that? When the thrombocyte count is more than 4 lakhs per cubic millimeter, we call it thrombocytosis. We will see when thrombocytosis is seen. Okay. So this under microscope, how they will look like. These are the thrombocytes, platelets. Okay, these are the RBCs and these are see the size. Much less than the RBCs. RBC size is how much? 7.2, on an average 7.2 micron. Then now say, what would be the size of the platelets? 2 to 4 micron. Much less. Okay, so this is the size of a thrombocytes or platelet. And how their morphology, how they will look like whether it is spherical or disc shape, what would be the shape? And the platelets, they are not having any nucleus. They are anucleated. Okay? Their 2 to 5 micrometers, their size, and the normal count is 1.5 to 4 lakhs of, as I said. They are formed in bone marrow, which says megakaryocytes. Mega means big. Karyo means nucleus. In the bone marrow, you will find, if you study the bone marrow, you will find the big cells are there. They are nothing but megakaryocytes. From these, the lymphocyte platelets are formed. Very simple way these are formed. Okay, and they are what is their lifespan? Short. It is 8 to 12 days. 8 to 12 days is their lifespan. And when they become dead, they are removed by scavengers. Who are the scavengers? Macrophages are the scavengers. Okay, where you will find most of the scavengers? In spleen. As we have seen, the senile RBCs are taken up by the scavengers, macrophages. So here also, one after their death, they will be removed from circulation by macrophages. The structure. They are constituted, the cell is constituted by cell membrane, and after the cell membrane, there will be microtubules. And these microtubules made up of tubulin, they actually just outside the cytoplasm and inside the cell membrane, these microtubules, like you have faced one question in physiology, what are we have to write short notes on microtubules, isn't it? The answers, most of the answers are very funny. So anyway, microtubules are there, microtubules, okay, and there is cytoplasm. Okay, so let's see what is there in cytoplasm. The cytoplasm is having a lot of things. What are this? There is presence of extensive canalicular system. That means the cell membrane, canalicular, the cell membrane, suppose this is the cell membrane, and this cell membrane becomes folded and enters within the cell. So these are the canaliculi. Canaliculi. What is the function of this canaliculi? Because the cytoplasm will having various types of, particularly alpha granules and dense granules, and they contains various substances. When these cells become activated by any means, by some means, when these cells become activated, they are degranulated and their contents are released into the canaliculi, then it comes out. So that is the function of the canaliculi in the thrombocytes. So extensive canalicular system is there. Microtubules provide the structure shape of the thrombocyte. Supports to maintain the disc shape. I asked, what is the shape of the disc? And when they become activated, the shape is changed because of they are having different type of contractile proteins like actin, myosin, thrombostatin. So these are the things they contract. These proteins can contract because of the contraction. And moreover, another unique thing, these platelets can also produce small pseudopodia. Okay, so we'll see when they become activated, they form spikes. These are pseudopods. Okay, and these are also essential for clot formation. Okay, and so The cell membrane contains lipids, various types of lipids. Cell membrane contains various types of, one is glycolipid, GP. You will find the cell membrane contains various types of receptors. And these are mentioned as GP1, GP1B, GP1A, GP1B, GP2A, GP3. So, the various forms of receptors are there on the surface. They are made up of glycoprotein. Moreover, there is glycolipid. There is cholesterol. Not only this, there are carbohydrates. Okay, carbohydrates, there are different classes of carbohydrates. One carbohydrate is glycocalyx. These are present exclusively on the cell membrane. So, carbohydrate is also present on the cell membrane. And what type of carbohydrate? It is glycocalyx. And moreover, there are some proteins. Proteins, enormous function. Here, within the cell, there are a lot of proteins. On the cell surface, there are also some proteins. They will be performing various functions. Okay, some will be performing as clotting factors. Some will be acting as enzymes. Some will be acting as receptors. Okay, some will be acting as secretory proteins. So, this protein has got various functions. You know, this glycoprotein, as I said, this glycoprotein, when they flow, these platelets, their outer membrane contains glycoprotein. Now, this glycoprotein, when it travels through the blood vessels, okay, the inner membrane of the blood vessels is made up of endothelium, and this endothelium is smooth. So, when the endothelium is smooth, these platelets do not adhere to the wall. They don't adhere to the wall. Okay, so they are actually they don't like to adhere there. Okay, so but if this endothelium by some means is damaged, then this glycoprotein will immediately bind with the damaged endothelium. Normally, they will bind. They will not bind. But when the endothelium becomes damaged, then they will bind with this. Okay, and it is called platelet adhesion. And when the wall of the blood vessel is damaged, endothelium will be damaged. At the same time, in the subendothelial layer, there is collagen. And this collagen is very active in binding the platelets. And the binding of platelets with collagen actually starts the process of hemostasis. So initially, what should be the first thing? The adhesion of platelets to the collagen, to the endothelium, with the various receptors that are present on the surface of the platelets. Now the membrane, lot of membranes are there on the surface of the platelets. And those particularly who are involved in hemostasis, what are these? Collagen receptor. And these collagen receptors are GP, means glycoprotein. Glycoprotein means what type of chemical it is? Glyco means carbohydrate, protein to protein. So this is a conjugated protein. Protein, when combines with a non-protein, it is a conjugated protein. So here non-protein substance is carbohydrate. So it is a glycoprotein. So it is expressed as G_p. So various forms of glycoproteins are there. 1A, 1B, 2A, 2B, 3A. So various types of glycoproteins are there. They act as receptors. So the collagen receptors, with this is the platelet. On the surface there is collagen receptors. So with this receptor, it will bind with collagen. It can directly bind with collagen, but in most cases, they bind with VWF. What is VWF? Fond, VUL, fond. W is Willeward, and F means factor. Fond, Willeward, factor. This factor is essential for binding the And this absence of this factor creates a disease. What disease? Platelet plug is not formed. Because for formation of platelet plug, platelet has to be linked to the bound, has to bind with the collagen with the help of this VWF, okay, von Willebrand factor. So with this it is written there, von Willebrand factor receptor, it binds platelets to endothelium via VWF, plays important role in platelet addition. Fibrinogen, there are fibrinogen receptors. We'll find how these fibrinogen receptors are essential for aggregating lot of seed. When platelets are gathered together, they are called aggregation of platelets. And how these are formed? Because you have some receptors. With this receptor, you can bind to one another. But you cannot bind to one another directly. In between, somebody should be there. So they will help in binding the platelets together. What are these? Fibrinogen. So in the plasma from the cells also, fibrinogen is released. But in the plasma, a lot of fibrinogen is there. Plasma protein. So this fibrinogen molecule attaches with one platelet. On the other end of the fibrinogen binds with another platelet. In this way, all platelets come together. This is called aggregation of platelets. Okay, so we will use different types. Adhesion or adherence of platelets to the endothelium, damaged endothelium. Adhesion of platelets to the collagen. So this is adhesion is one thing. And after adhesion, the platelets become activated. When they become activated, they are granules. The cells are having granules. These granules will be secreted. Not only this, they will change their shape because the cell is having a lot of proteins, contractile proteins. With the help of this protein, they will shape their change. They will produce spikes. Okay, so these spikes will help in aggregating you. Then there will be aggregation. When there will be aggregation, then platelet plug will be formed in your area. As a result, there will be no more oozing of blood, no more hemorrhage from that point. So it is a temporary plug. Temporary. This is not that fibrinogen with which the platelets are attached. No, the platelets are attached to one another by fibrinogen. This fibrinogen is soluble form. These are soluble. See, if I can make it insoluble, then the total plug will be permanent. And this is done by in phase 3 with the help of intrinsic and extrinsic pathway, which we will be discussing in our Saturday's class. So there will be collagen receptor, there will be von Willebrand factor receptor, there will be fibrinogen receptors on the platelet surface, there will be ADP receptors, ADP. See, this ADP is also very much essential. It binds to amplify the platelet activation, aggregation, shape change, and release of granules. So ADP is also essential. ADP is released from the granules. Okay, so this ADP will bind to the ADP binding sites, ADP receptors, and they will cause various changes in the platelets, like activation, aggregation, shape, change, release of granules. Okay, so another receptor is there, thromboxane A2, T suffix X, A2. So this thromboxane A2, like this, thromboxane A2 receptor, this receptor binds thromboxane, release from active thrombocyte, activates other platelet aggregation. Means when they are released, this platelet aggregation, everything is aggravated, amplified. Only purpose is to seal the damaged area so that bleeding is stopped. Okay. Now the cytoplasm. In the cytoplasm, you will get actin and myosin. Contractile, these are contractile protein, and they maintain the spherical shape of the platelets, and they also produce pseudopod, or that very spike-like structures. Okay, thrombostatin is another protein present in cytoplasm. This is also like actin and myosin. They are contractile protein. And what is their function? Which can cause platelets to contract. I said, on a clot retraction, we'll find its importance during that time. Clot retraction. Blood vessel, this part was injured. So this part there is clot formation. First there will be plaque formation, then it will be clot. Okay. So now what will happen? This clot will be retracted. When this clot is retracted, so they are having this thrombostatin. Because of this thrombostatin, each of the platelets will shrink. But they are tightly adhered to one another by fibrinogen, insoluble fibrin. So as a result, what will happen? They will bring the endothelium closer, because you are attached to the endothelial lining, she is also attached to the endothelium. So when you will be shrinking together, so both the end of the endothelium will be coming closer. Okay? Then what will happen? This will be fragmented, and will be taken up by the macrophages. And by this time they are coming closer, and by this time another factor is released from the platelets, and these platelets causes cell division of the endothelium, cell division of the muscles, cell divisions of the connective tissue. So as a result, what will happen? They will proceed, they will cover the damaged area, there will be tissue repair. Okay, once this tissue repair is done, hemostasis mechanism is done. Okay, so this is a very interesting one and a complicated one also. Okay, so thrombostatin, beside you will find endoplasmic reticulum, you will find Golgi bodies, you will find mitochondria, these are there. Okay, these are vesicles and the enzymes, lysosomes, these are present there. And more importantly, there are two types of packets of granules. One is dense granules and the other one is alpha granules. What are the constituents of dense granules? These dense granules contain non-protein substances like glycolipids, phospholipids, cholesterol, and these are mostly coagulation factors. 5-hydroxytryptamine, and it is a vasoconstrictor. It is a vasoconstrictor, and it is other name of the 5-hydroxytryptamine is serotonin. They can create spasm in the vessel. Okay, suppose this area is damaged, so immediately, reflexly, there will be spasm. Okay, and you will feel pain. This pain, reflexly, reflects by reflex mechanism called axon reflex, AXON. Through axon reflex, it will cause local vasoconstriction. One is reflexly done, another is, when damage is done, myogenic reflex. No need of any neurons or any nerves, only by themselves. When the wall is damaged, when the muscles are damaged, smooth muscles are there, immediately there will be constriction of the smooth muscle. So there will be narrowing of the vessels. Moreover, what will happen? Moreover, the pain will be created here. This pain will cause reflex vasoconstriction. Moreover, when this is damaged, some platelets will be activated. And these activated platelets will secrete something like 5-hydroxytryptamine, thromboxane A2. They are endothelin. They are strong vasoconstrictor. So before coagulation, hemostasis started, platelets aggregation started, actually this process, vasospasm or vasoconstriction, it is the initial stage of hemostasis. So what is the initial stage? Vasoconstriction. 3/3 means there may be vasoconstriction. One is myogenic reflex, another is action reflex, another is the platelets which will be activated, they will be releasing some substances like 5-hydroxytryptamine, like thromboxane A2, like ADP, they will cause vasoconstriction. So by 3 means there will be spasm at the first stage, and purpose, if the vessels is constricted, there will be less flow of blood, less loss of blood, and hemostasis means prevention of loss of blood. Hemostasis means prevent the mechanism by which loss of blood is prevented. So thereby, by this phase, first phase, we can minimize the blood circulation, blood loss. Okay. So these are the contents of Grange granules. And what are the contents of alpha granules? See, what are these clotting factors and von Willebrand factor, VWF factors. These factors are essential for platelet adhesion to the site of injury. Fibrinogen, I told, you will be linked by fibrinogen. So they are also released from here. Factor 5, it will be needed in third phase. And thrombospondin, what is this? It promotes platelet aggregation and hemostasis. Upon platelet activation, TSP is released from the platelet, alpha granules, where it can make a bridge between the platelets and stabilize fibrinogen. Okay, so it actually stabilizes the fibrinogen. Okay, these are fibrinogen loose, no, they are soluble. So its function is to make it stabilized. Okay, platelet-derived factor. This platelet-derived growth factor, its function is the last step, tissue repair. Damaged endothelium to repair of the web. So its action will be at the last stage. So these are the factors released from the alpha granules. Now, this has these are a big different thing, canaliculi, and this is the alpha granules, these are the beta granules, lysosomes. So, dense tubular systems, everything. This is one of the glycoprotein receptors. This is the fibrinogen. So, another platelets will be having glycoprotein, and with that it can bind with this fibrinogen. So, two platelets can come together. This is done during aggregation. Okay, so platelet is required to bind both of you in between platelet. You will bind fibrinogen, she will also find fibrinogen. So you will be coming through binding, you will be coming together. This is aggregation. Okay, structure over. Now leukopoiesis. Sorry, not leukopoiesis, thrombopoiesis. What is thrombopoiesis? The process by which thrombocytes are formed. If you study the smear of bone marrow, what you will find? You will find big cells. These are the big cells. See, and these are the cells. So these are cells. Other cells are like this, whereas these are very big. That's why they are called megakaryocyte. The platelets are also formed from megakaryocytes. How? Very simple. This megakaryocyte produce uropod, produce extension, or you can say roti. जब आटा mix करते हैं ना with water, then you make a dough. You make a dough, then long dough, then you do it like this. Here also. The cell with some, you will pinching. From outside, you pinch some portion of the cell membrane along with cytoplasm. Okay, so then tear it. So this is one platelets. One thrombocyte, one megakaryocyte can produce 2,000 to 4,000 platelets. Only one megakaryocyte. How many platelets it can produce? 2,000 to 4,000 platelets. Okay? And what is the process? So, SSM is committed pluripotent hemopoietic stem cells. Origin is same. All blood cells are originated from hemopoietic stem cells. So, these hemopoietic stem cells will multiply and another variety of cells that are committed to produce megakaryocyte will be formed myeloid progenitor cells. Now they will divide and differentiated into megakaryoblast. I always tell blast means big size and their mitotic capacity is very much. So it will be formed megakaryoblast. Then from megakaryocyte, pro-megakaryocyte will be formed. See the changes in nucleus. Pro-megakaryocyte will be. Now this will be changed into megakaryocyte. And from this megakaryocyte, there will be pinching. Platelets will be formed. So this is what? These are the different stages of. It is only one term can be used, but different steps are not usually asked from actually. It is asked for the step erythropoiesis. You will face questions from erythropoiesis, different stages. Okay. Now the functions. So what is the first function of this? Blood hemostasis. What is hemostasis? Cessation after injury. Cessation of blood or cessation of hemorrhages. Okay, loss of blood. Cessation of loss of blood. So how it has got various phases. First, we have seen vasoconstriction. Then what is the platelet adhesion to where the platelets will be adhered in the collagen, damaged collagen, and in the damaged endothelium. Okay, so this is addition. Then platelet activation. When they will be attaching this collagen, they become activated. And when they become activated, they change the shape. They secrete different substances, granules present in these dense granules, alpha granules. That will be secreted. Then, as a result, there will be platelet will be aggregated. Okay, so aggregation and there will be formation of platelet plaque, and this one is temporary platelet plaque. Temporary hemostatic plaque, stoppage of bleeding through injured blood vessels. Then blood coagulation, the final step. It also involved in blood coagulation. What is coagulation? Coagulation is the process, remember this. Coagulation is the process by which blood is transformed into semi-solid, jelly-like structure. So this is called coagulation. The fluid blood is transformed into semi-solid, fluid-like structure. So by the process of coagulation. So it also helps in coagulation. Clot retraction, it also secretes something that causes clot factor 13, it causes clot retraction. It has phagocytic action, particularly in the blood, there is antigen-antibody reaction and complex. This complex can be engulfed by them using their pseudopodia. So that is another function. Since they contain in the granules lot of 5-hydroxytryptophan, so they are storage of this substance, particularly during allergic reaction. These are released and allergic manifestations is produced. and repair of injured blood vessels. So all of these, there are 6 different functions of platelets. Okay. These functions is asked, and you can face this question. Okay. Now they count when they can be count can be decreased, count can be increased. So decreased means thrombocytopenia. What are the conditions in which it is decreased? So these are the conditions. Bone marrow depression, hypersplenism. When the spleen of your body becomes enlarged, so you will find in the blood cell, this count of platelet is also reduced because they trap it and destroy it. Okay, and there is in viral infections, in HIV infection, in splenomegaly, drug hypersensitivity, and irradiation of bone marrow. So there will be lack of functions in the bone marrow, so their count will be reduced. There will be thrombocytopenia. And thrombocytosis, when epinephrine is administered, its amount is increased. And after trauma, surgery, injury, that increases thrombocytosis, that increases platelet count. Splenectomy spleen is removed, so there is nobody to destroy the platelets, store the platelets, so their count will be increased. Okay, stress increases this count. Chronic infection also increases the count. Okay, so let us now coming into hemostasis. I defined already what is hemostasis. When a blood vessel is damaged, the very first response is rapid vasoconstriction. By 3 means, I already told you, by 3 means there will be vasoconstriction, and the purpose is reduce the blood flow, reduce the loss of blood through the damaged area, and this will be followed by platelet addition, then platelet activation, then platelet aggregation, and then formation of platelet. So these are the sequence of events. 5th row, second boy. Yes, 5th to second boy. What are the stages? Okay, then what will be this is phase 1. Then there are how many means by which there will be vasoconstriction? How many means? 3 means. One is myogenic mechanism, another is action reflex created by pain receptors, another is that active when the platelets will be passing through the damaged area. What will be there? They will release the granules, and this granule contains 5-hydroxytryptamine, thromboxane A2, they will cause vasoconstriction. Okay, these are the 3 means. Okay, first phase gone. Then second phase. Second phase involves all this. Starting from here. So when it is over, then there will be platelet addition. Platelet addition to where? Damaged collagen, exposed collagen, and exposed damaged endothelium. Okay? Then what will be there? When these are adhered to that part, that collagen and endothelium, the platelets will be activated. As a result, platelet, there will be aggregation, there will be secretions of granules, and they will cause aggregation of platelets, and then there will be formation of platelet plaque. So these are the sequence of events that take place in platelet plaque formations. Okay, so see, these are the blood vessels, and what is there? This is the endothelial layer, endothelium, and this is the subendothelium. Here you will find collagen. Here is the muscular cord, and here is the tunica adventitia. Here also you will get collagen fiber. So into the subendothelial layer and also in the tunica adventitia, outermost layer, here you will get endothelium, collagen, and the innermost layer is endothelium. So here the platelets can adhere in the damaged part, and also here they can be attached to the collagen fibers. But if it is smooth, here no platelet will be adhered. Since it is smooth, they will repel. They will repel adhesion. Okay, and they are having endosthenin. With this endosthenin, they will repel. These are the phases. So what is this? When an injury is made, there will be exposure of collagen, and von Willebrand factor will be coming, and with this, the platelets will be adhered to the collagen. Now the platelets become activated. Now three phases start. One is vasoconstriction, secretion of serotonin, 5-hydroxytryptamine. This will cause vasoconstriction, stage 1. Then what will happen? They will adhere to, with the help of thromboplastin A2, they will adhere to the collagen fiber, and there will be aggregation of platelets, there will be formation of plaque. This is stage 2. And in stage 3, what happens? I didn't describe it. I will describe in the coming classes how it happens in stage 3. Okay, so next, I already told. First event is vascular constriction, stage 1. Then formation of temporary platelet, stage 2. Formation of blood clot, stage 3. Then there will be clot retraction. Then there will be tissue repair. Okay. So stage 1 done by local myogenic response. So this is the damage. So reflexly, these are constricted, blood flow is minimized. Okay, then what is there? Then there is release of vasoconstrictors from the endothelium and platelets. Say these are what? Thromboxane A2, serotonin, endothelin 1, they cause vasoconstriction. It is a chemical induced vasoconstriction. Okay, and the final one is reflex initiated by pain receptors, spinal reflex, this is action reflex. Because of this action reflex, there will be vasoconstriction. As a result, what will be there? There will be reduced blood flow into the injured place. Then comes the final second stage. Second stage is formation of platelet plaque mechanism. First platelet addition, then platelet activation, then platelet aggregation, formation of hemostatic plaque. Okay, so these are the stages. And see, these are the collagen fibers, and this collagen, because of the damage of this endothelium, collagen is exposed, and now there's these cells, and these cells, they secrete this substance. So this substance, that is the von Willebrand factor, they binds the collagen. The other end of this factor binds the GP1B receptor of platelet. As a result, this platelet becomes activated. Okay, now when the platelet become activated, what will be the response? They will form spikes. They will secrete several substances. With that, they will produce aggregation of cells. So when platelets are activated, ADP, thromboxane A2, serotonin, calcium, these are released. And all of this will help in aggregation of the platelets. See, these spikes are called philopodia. What is philopodia? Spikes, pseudopods, small spawns, extension produced in the platelets because of activation of the platelets. So this spiky extension of platelets is called philopodia. So philopodia is formed. They secreted the granules. So with these granules, now what happens? See, these are the pseudopod or philopodia. From the philopodia, fibrinogen is attached. This fibrinogen binds another. It will be with this lot of fibrinogen, lot of platelets will be adhered together, and a plaque will be formed in this region. So with all these receptors, these are the collagen. What are the receptors? They actually cause platelet activation. And what are the receptors? By which? See, this is the GP2B and B2. So this is this receptor. It binds fibrinogen. other end of the fibrinogen will bind with platelets of another GP2, and with this a link is formed. So when platelets are active, shape is changed, release of granules and amplification is made, and platelet aggregation, there is actually these platelets are having GP2A. When the platelets are activated, these are transformed into GP2A, 3, 2B and 3A. Okay, now because of change in these receptors, now they will be able to bind with other platelets with this receptor. If these receptors are not, they will not be able to bind other platelets. So this transformation is essential. This receptor is essential. So this transformation is made, and because of this transformation, fibrogen bridge will be formed, and release of the thromboxane A2 increases. This increases when these are activated. Thromboxane A2 is formed, and it will increase the platelet aggregation. Von Willebrand factor released from the damaged tissue. It combines with GP1B receptor of the platelets. Now this platelets is now adhered to the collagen. Now this is the platelet receptor with which it will be binding with the fibrinogen. The other end of the fibrinogen will bind with another platelets. So in this way, a lot of platelets will be aggregated. So that's all. This platelet plug is formed. Okay, so there will be oozing will be stopped, or bleeding will be stopped, hemorrhage will be stopped, and then the next part will be studying how this temporary plug is transferred into permanent one. Then how it is broken down, retracted, and how growth is made.